Well, got it. Well, good afternoon or good morning, wherever you are today. It's Russ Read speaking, and I'm the Executive Director of the National Center for the Biotechnology Workforce located here at Forsyth Technical Community College, Winston-Salem. And I'm also a co-PI in AAV Bio, and we're so happy to host the WEBET group today. I'm a part of this project, and WEBET stands for Workforce Expansion in Biomanufacturing Emerging Technologies. This is part of NIMBLE, which is the National Institute for Innovation in Manufacturing of Biopharmaceuticals. It's project 2.2-152, and NIMBLE is one of the 16 Manufacturing USA institutes, networks of public-private partnerships that work with academic and private sector in the manufacturing organizations for research and development of manufacturing skills training. So today we're really excited because we have a project that's come to fruition, and this is the second in the series of our training for you. So Maggie, if you can go to the next slide, please. First, I'd like to introduce the team. So the WEBET Partner College and Innovation team is Montgomery Community College in Bluebell, Pennsylvania, and it's Dr. Maggie Bryans, who's the co-lead with Hitel Dashi. Solano College in Vacaville, Jim DeKloe is our co-lead. Myself, Russ Read at Forsyth Technical Community College. Iso Bale at Quincy College in Quincy, Massachusetts, in the Massachusetts Life Sciences Center. And Dominic Ingato and Barbara Jacunso, co-PIs from Miracosta in lovely Oceanside, California. And Louise Petrella, who's now with Biomed, is a co-PI, and she's with Shoreline Community College in Shoreline, Washington. So just on the side here on the right, you can see that the top US biopharma clusters for 2021 are Boston, San Francisco, New York, New Jersey, Maryland, Virginia, DC, San Diego, Los Angeles County, and Greater Philadelphia and Seattle, Washington, and Raleigh-Durham, North Carolina, and the Chicago region. So just as we go into the next slide, you have to understand that we did a labor market analysis of these particular sites so that we can really understand what are the needs in this particular market of cell and gene therapy. Next slide, please. So here's the agenda for today. Welcome and introductions, thanks. And the learning module for cell and gene therapy from Dr. John Cumbers. Thank you, John, for joining us today. Update on CGT skill standards with Maggie and Jim. A brief overview of CGT and AAV production with Jim and Maggie. CGT analytics and microbiological control with Jim and Maggie, regulatory considerations with Jim, then we'll take a break, and then we'll do lab-based curriculum analytics for AAV transduction with Dominique and Maggie, and then analytics AAV production with ISO, and then we'll do equipment necessary to train CGT technicians with Barbara. Thank you. Next slide, please.
Thank you, Russ. This is Jim DeKloe, and I have to say this has been a fascinating project that when we started, we guessed that cell and gene therapy would be hot. And in the interim, in the several years that we've been using this project, there are more and more companies, more and more therapies, and it just has been so interesting. And also, many of you are considering or have launched a cell and gene therapy certificate. It's my pleasure to introduce my friend and colleague, John Cumbers. John, I met, he's the founder and CEO of SynBioBeta, which is an advocate for synthetic biology. Recently, he has a new endeavor where he's developed a new learning platform called LalTal. And LalTal has a learning module for cell and gene therapy, which I've been working with and I've found to be fun and very useful as a way to learn cell and gene therapy. So let me turn it over to John, and thank you, John, for coming today.
Absolutely. Thanks, Jim, and thanks, Maggie, Russ, and Sandy for letting me talk to you about LalTal. It's good to meet everybody. I'm really passionate about synthetic biology and even more passionate about cell and gene therapy and the potential that it has. I've reviewed the course materials from the first workshop and really enjoyed all of the presentations. However, I know how difficult the jargon in biotechnology can be, and particularly with cell and gene therapy. It's a very complex topic with many terms that are complex, that are difficult to understand. I've been developing over the last 18 months a tool to help teach people biotechnology and other deep tech topics, and I would like to share it with you. Jim and myself have been working on a series of questions about cell and gene therapy that will allow you to deepen your understanding of the topics. In particular, the way that LalTal works is it allows you to map out what you know, what you don't know, and then it connects you with other people via a voice memo, asynchronous audio app, where you can connect and test your understanding of these different topics. If you get it right, you get a thumbs up. If you get it wrong, then somebody will reply to you and help you learn. It's a very fun, interactive, engaging environment. Jim and Sandy and Todd were all on an event that we did last week on cryptocurrency. And a lot of people really find it an interesting and fun way to learn. So I'm going to host a follow-up event to this workshop, either this Wednesday at noon, this Friday at noon, or next Monday at noon. And I've just posted in the chat here a short survey. So if people are interested in trying this app, showing and reviewing and deepening their understanding of cell and gene therapy, then please click the link, tell me which day would work, which day and which time would work best for you to join an event, and I'll follow up and send you a link where you can install the app and see how it works. Previous attendees have really found these a lot of fun and a really engaging and fun way to learn. So I think that you'll enjoy it too. So thanks again, Jim, and thanks to everybody for letting me talk to you about LalTal and what we're building.
Thanks so much, John. And thank you for developing that cell and gene therapy component of this app.
Yeah, I learned a lot too, so it's a lot of fun.
Thanks.
Thank you, John. I look forward to looking into that for sure. So next, Jim and I would like to give you an update on the skill standards that we developed in the WEBET project, the whole team. And we have actually developed a report which should be finished this week and be published on the MBC2 and Innovate Bio websites. So we'd like to just show you some of the outcomes of that whole effort. And it really started with one of the first activities we did with this project was to host 6 regional SME listening sessions. So each of the 6 colleges brought in their cell and gene therapy industry partners. And before they even came to the table, I think Russ and Izzo both had theirs face-to-face, which was great because they snuck it in just before the pandemic. The rest of us had virtual sessions. But before they got together, before the group got together, we gave them the NBC2 traditional biomanufacturing skill standards for manufacturing and QC technicians. And we asked them to review them and then come back and let us know what was similar for cell and gene therapy manufacture and what was different, and to highlight those differences so we could really build some skill standards that were specific to cell and gene therapy. And these are industry partners. It's quite an impressive bunch, I think, and many times more than one SME came from each company. So we really had a good representation from our partners. So there were 27 companies and 3 educational institutions that came together to help us develop these skill standards. So with the help of John Carisse, we put that together in a report, and this is the cover of our report, Skill Standards and Curriculum for Cell and Gene Therapy Technicians. And as I mentioned, it'll be ready by the end of the week for everyone to view. And this is just sort of a picture of the table of contents. So from the work that we did with our SME partners, we developed skill standards that are common for both cell and gene therapy technicians. And most of the critical work functions and many of the key activities were common across both products. And of course, many were in common as well with traditional biologics, protein biologics. And then there were some skill standards that were very specific to cell therapy and gene therapy because they are different products. And then we also asked our industry SME partners, if we were developing a course in cell therapy or a course in gene therapy, what topics should we include? And we get a wealth of information on that as well. Foundational knowledge, specific skills, biomanufacturing knowledge. So we included that in the Skills Standards report. That's Curriculum for Cell and Gene Therapy Technicians course curriculum. We also asked them what non-technical skills these technicians should have coming in, and how could we prepare them and fold those non-technical skills into the courses. So we have a great list of those skills. And then lastly, and Barbara's going to talk about this today, what are the recommendations for equipment? What equipment should we purchase in order to be training these cell and gene therapy technicians? So I think all in all, it's about a 28-page booklet with a lot of great information. And I put the link here. It's not on the website yet, but this is the link at the MBC2 website under manufacture of cell and gene therapies, where you'll find the skill standards document. And Jim's going to say just a little bit more on the layout of the report. Jim.
Also at that site is our labor market information, and that shows that this field is increasing exponentially, and there'll be a lot of opportunities for your students to get in on the ground floor of this field and to move up, because it has a tremendous ability for them for upward mobility. Many of the subject matter experts came out of what we're calling traditional biomanufacturing. They were producing protein biopharmaceuticals, and then we asked them, in your new job, what is different? How does this differ from what you were doing before? And just to point out a few of these, the product is a cell or a virus, much more complicated than a single protein. There is a lot of overlap with traditional biomanufacturing, but there's a higher sense of urgency. You're on the clock, especially for CAR T therapies, where the patient can't wait. The patient is going to die in a month, and you have to as quickly as possible, produce that therapy to get back into the patient to have the CAR T cells attack their cancers. So there's a timing difference. There is a scale difference. You scale out rather than scale up. And for us, this is actually a little bit of a benefit, because the scale is closer to our laboratory scale than it is to a giant 25,000 liter bioreactor. And so we were looking at it. The raw materials are much more variable. And really a big difference is that you can't terminally sterilize. You have to maintain sterility throughout the entire process. So what skills are required? Aseptic technique, aseptic technique, aseptic technique. If your students can work in a biosafety cabinet, if they can transfer materials from here to there without getting them contaminated, if they can work with sterile welders and sterile connectors, then they will always be employed. Foundational knowledge, immunology, which is somewhat different maybe than, the emphasis is different than we had in the past, cell biology and virology. And in my other class, I didn't talk much about viruses except as something bad and to avoid. In this case, they are the product. And so you have to nurture the viruses and love them for both of them. And so to summarize, we collected all of the input from the subject matter experts, and every session was fascinating and gave us gems from the industry point of view, and we put it together into this report. And again, as Maggie said, we are looking forward to, it's almost done. We'll have it published by the end of the week. Thanks, Maggie.
Thank you, Jim.
Next slide, please. So we thought that in the first session, we spent 2 hours talking about cell and gene therapy and what is it. And we thought that it might be good to take 5 minutes or so and review it for anyone who may not have attended that session. So cell therapy, where the product is a cell rather than a protein. And this is different in a lot of ways, although we have a long experience with that. We have been transfusing blood for over 100 years and matching it to blood types, and we have been doing we've been doing transplants, and that is cell therapy. Tied in with this is the emerging field of stem cells and implanting them. But in this case, CAR T is along, and there are a few approved products, but hundreds of products in the pipeline. In gene therapy, you manufacture a virus, and the virus then is a carrier for the gene that you would like to introduce into the patient's cells. You inject the viruses, and if you're familiar with virology, the numbers are really big. A typical dose is 10 to the 15th viruses, and the viruses then go through the bloodstream. Often they're targeted for a particular tissue, and they introduce the DNA into that particular tissue. Next slide, please, Maggie. Before that, yes, thanks. So we're talking about, in the past, autologous CAR T cell manufacturing. And this is sometimes called vein to vein. And in traditional manufacturing, you have vial to vial, from a vial of cells through production to a vial of protein product. In this case, it's vein to vein. The cells are removed from a patient through leukapheresis, and then they're frozen and mailed to a manufacturing plant. At the manufacturing plant, they'll insert the gene for the chimeric antigen receptor, and typically they use a lentivirus vector in order to do that. That information then will be used by the cell to display on the surface the chimeric antigen receptor, which has a business end that is specific for binding the cancer that is the target of this. And currently, all of the applications are types of leukemia. It isn't sure that it could work on solid tumors, but everyone thinks that it does. Then those genetically engineered cells are activated, and they divide. That's where it takes most of the time. Then at the end, they'll be frozen, mailed back to the physician to go into the patient, and they'll run around the patient's body, destroying cancer cells. And these results are very often spectacular. People are talking for the first time the C word, a cure for cancer. Next slide, please. Slightly different approach for allogeneic CAR T cells. And this has a prospect of producing CAR T cells closer to the process that we use for traditional biomanufacturing. In this case, you take the white blood cells and you collect them and select for them out of a healthy patient. You activate them, genetically modify them, again, typically with lentivirus, and then expand them up in a larger bioreactor, and then freeze them into different doses. So in the previous example, one cell is expanded from one patient and goes back into that same patient. In the allogeneic approach, one person donates cells, and in this case, it's a healthy person, and the cells are genetically manipulated not only to display CAR T, but also to not attack the patient in graft versus host disease, and to not be rejected by the patient as well. So again, in this case, you can bank doses of these and then defrost them and give them to a patient for the therapy. This approach is less far along than the previous approach that we talked about. Next slide, please. And the way that this is shaking out, there are a lot of different viral vectors that you can use, but the way that it's shaking out is most companies are using lentivirus to transduce their T cells to produce CAR T cells. And most companies are using adeno-associated virus to produce their gene therapies. Lentivirus is a retrovirus that has an RNA genome. Adeno-associated virus is it requires a helper virus, that is, it cannot cause disease by itself. And it's a single-stranded DNA virus. In both cases, you take out the guts of the virus and put in your transgene, and then the virus is merely a way to introduce information into the cell. In cell culture, you can grow the cells as adherent cells, as shown here in these stacked bioreactors, or you can adapt them for suspension and have them produce viruses in that way. Next slide, please. So the production, and ESO is going to talk about this, but often you can use HEK 293 cells, human embryonic kidney 293 cells, as the host cell for production of lentivirus. These cells are also used to produce adeno-associated virus. In addition, there's an alternate. You can use the insect cells, SF9 cells, to produce lentivirus. And this shows the sequence of events, and ESO is going to cover this. Next slide, please. Upstream production, you harvest it, and often when you harvest these viruses, you will break them open with a detergent that spews the host cell DNA in there. So you'll add a deoxyribonuclease, a DNA ACE that is commercially available to digest the outside cellular DNA. You clarify, and then in the case of adeno-associated virus, you use an affinity column that has a camelid antibody that's specific for the particular capsids of the serotype of the strain of adeno-associated virus you have. You capture it, and it's typical chromatography where the impurities go through, and then you release the virus, and then often there is a ion exchange chromatography step there in order to polish it. Next slide, please. And Maggie, this is you.
Thank you. So when we were deciding on developing curriculum, hands-on curriculum for the WEBET project, we understood that we'd be using HEK 293 cells, and Izzo took that on, and he has been producing recombinant AAV and HEK cells. But we also wanted to work up the SF9, the insect cell production as well, largely because these cells are biological safety level 1, and we understood that maybe a lot of programs would like to have a cell line that was BSL1. And when we talked to our industry partners, they said both would be very relevant. So Issa will talk later about HEK as HEK 293 cells, as Jim said, but Jim and I and Hetal have been working up a process for using SF9 cells, insect cells, for AAV production. So SF9, Spodoptera frugiperda, moth insect cells can be used to produce recombinant AAVs. So similar to using baculovirus expression in insect cells for protein production. I think one of the COVID-19 vaccines from Novavax was produced, spike protein was produced using baculovirus system in insect cells. So that's been around for a long time. So it was developed then to be able to produce AAVs. And the way it's done is that the SF9 cells are infected with two baculoviruses, two recombinant baculoviruses. One has the gene of interest. In our case, we're hoping to use GFP, and the other one has the AAV helper genes, the REP and the CAP genes that allow replication of the virus, and the capsid proteins allowing packaging of the virus and the viral particle to be made. And this insect-based systems actually has been used for a product that was approved and is no longer on the market, Glybera, which was gene therapy used to replace a lipoprotein lipase gene. It was approved in Europe, but not in the US. But here's your SF9 cell here. They grow in suspension. They're fairly easy to grow. And the two baculoviruses that you co-transduce the cells with, and then the cells will make the recombinant adeno-associated virus. So we're hoping to get this system up and going. We've had some roadblocks, some hurdles along the way, but we have some good ideas about how we can get this production running. So SF9 cells, again, we can purchase them from the ATCC, or Thermo Fisher has its own sort of a clone of SF9 cells called XP SF9 cells. They're biological safety level 1. They grow in specialized insect cell medium. There's a couple different ones out there, and Jim has a good contact near him that actually a company that produces the media. They grow at 27 degrees, so a little bit warmer than room temperature at 125 RPM. So we grow them in shake flasks, 125 ml shake flasks, 250 ml shake flasks, for example. And you passage the cells when they reach a density of 5 to 10 million. So these grow at a higher density than HEK cells. They have the potential to actually produce more AAV viruses than HEK cells. So you grow them in suspension. And as far as getting the recombinant baculoviruses, so the University of Florida has several publications, a group there has several publications out on a system that they have put together to produce GFP using a recombinant adenovirus with the GFP gene. And they actually also developed a special cell line, a packaging cell line. So the cell line is stably transformed with the AAV rep and cap gene sequences under the control of an insect cell gene promoter. So only one baculovirus is needed when you do the transduction. So you only need a single recombinant baculovirus carrying the GFP gene. So they have both the cell line and the recombinant GFP baculovirus. It's just, making the collaboration and filling out materials transfer agreement and perhaps being able to use this. But we thought Jim and I received permission to use that, we weren't sure the whole community would get the same permission, because they are now working with a company to work this system up. So we've been looking around for an alternative, and this system here is what's done at BTech, and also I think at in Texas Bioprocessing Center there. So looking around, we found that commercially available baculovirus, recombinant baculovirus, was available for GFP with the inverted terminal repeats, AAV inverted terminal repeats. So that's one of the baculoviruses that you need to transduce, and also the helper baculovirus. So we can actually purchase both of these baculoviruses from Vector Builder in a sort of a package of the two of them. They won't sell either of them independently. So the rep cap baculovirus will provide the viral genes for the replication and the packaging. So we have capability of purchasing these recombinant baculovirus and perhaps making our own stocks of those recombinant baculoviruses that we can use to transduce batch after batch of SF9 cells. So that's probably the route we're going to take. Working out, there's a lot of material out there as well. The process is fairly well worked out as far as cell density that you use to transduce, incubating with the correct concentration of baculoviruses, and incubation. So you do the transduction and you incubate for 3 days while the cells produce the AAV. Then you harvest the cells, like Jim was mentioning. The cells are then lysed using a lysis buffer, conveniently with the Triton X100 detergent. So it's a fairly easy process to collect the cells and lyse them. And moving on to sort of downstream processing after you clarify the lysate. So using the ActiPure system, this is a chromatogram here from the affinity chromatography that Jim explained just a moment ago. In our case, we're going to stick with the Thermo Scientific product, and they have a Capture Select serotype specific resin that does have that. So it's an antibody-based capturing the capsid proteins on the outside of the virus. And here's a chromatogram here from the GIBCO sort of a Thermo Fisher application note. You can see this is the flow through, the load of the lysate from the cells, then a few washes, and then finally the elution where the AAV is coming off of the antibody that's attached to the beads. In this case, this peak will contain both full and empty capsids. So in order to separate them, a polishing step with anion exchange is typically used. And this is based on the slightly different isoelectric points between the full and the empty capsid. And here's an SDS page gel just showing what was loaded onto the column, the flow through, and then the pure, here you're seeing 3 capsid proteins that we'll circle back to in just a moment, VP1, VP2, and VP3. So there's protocols out there, there's methods out there available for us to follow to grow the cells, make the AAV, and purify the AAV. So I'll pause there for a second and see if there's any questions. Maybe there's some in the chat. If not, we can move on.
No questions.
Thank you, Barbara. So we'll move on then to cell and gene therapy biomanufacturing and the analytics associated. So this slide has a lot of information on it, and getting back to what Jim said earlier, some of what's done in the analytics has similarities or is the same as traditional biomanufacturing. Assessing purity, for example, host cell DNA is done by qPCR, so that's an FDA approved method that's already been in the works for a number of years with protein biologics manufacture. Similarly with testing for host cell proteins, this is both drug substance and drug product using ELISA methods. Unique to cell and gene therapy is analyzing the ratio of empty versus full capsid. And we mentioned there that anion exchange can be used to purify full capsid away from empty capsids, but the analytics still have to be done. And this can be done by analytic ultracentrifugation, where they settle at different points in a centrifugation gradient. Transmission EM is a very popular method for looking at empty versus full. It doesn't give a direct quantity in that way, but you can have a ratio. Capillary isoelectric focusing is a method that's coming into play, as well as HPLC for separating both of them. And also sometimes just comparing and looking at the ratio of the information from digit drop PCR when you're looking at a number of viral genomes to the ELISA information from the capsid protein. So looking at protein versus DNA and that ratio to get a ratio of full versus empty capsids. Virus purity is also determined by STS page traditionally, as we'll see in just a moment, but also capillary electrophoresis, which is common in traditional biomanufacturing, HPLC, and analytic ultracentrifugation again. To quantify the number of viral particles, looking at viral genomes, Jim's going to explain in just a second, digital droplet PCR is the method of choice. It's quantitative PCR, but differs from the traditional real-time PCR that we use, and that will quantify the number of vector genomes. And then again, ELISA for total vector particles. ELISA specific to VP1, the capsid proteins VP1, 2, and 3, and the particular serotype of virus that you're purifying. Also quantifying infectious particles using a plaque assay or another cell-based system. Identity, confirming the identity of the virus and the viral serotype using antibodies against the specific capsid proteins, antibodies against, for example, AAV2 or AAV6, whatever serotype you're purifying. And this can be done using Western blot or LC-MS, liquid chromatography mass spectrometry. Of course, vector genome identity has to be done as well to ensure that the virus actually has the transgene and the correct transgene, and it is all as it should be. And that's done by Sanger sequencing or next generation sequencing. And then, of course, just like traditional biologics, potency tests have to be done. And these are typically a cell-based assay. Does the virus deliver the gene to the cell? Yes. Is the protein active? So activity acts to show that the gene is delivered, the protein is made, and actually is doing what it's meant to be doing. So all of them are done in a batch by batch method, of course. Or also an in vivo assay may be done during development. safety. So safety we'll talk about in a little bit. We'll talk about microbiological control, but safety testing for sterility, bioburden, endotoxin levels, and the presence of mycoplasma. And we'll talk about them in more detail in just a second. So those are some of the unique analytical methods to cell and gene therapy, and others, again, are very similar to what's done for traditional biologics.
Maggie, we did have one question. Someone is looking at the cost of the Capture Select AAVX at 7,500 for 50 milliliters and was asking if there might be alternatives that are cheaper for community colleges.
Yeah, there are a few others. It's about the same price as Protein A. So we would typically buy a 1 mil column, and that's about $100. So I think keeping the scale small, we'll be doing this in small scale. I agree, it's very expensive. Those affinity resins are expensive. There are 2 or 3 different ones out there. I think Cytiva also has a resin, that maybe that's what ISO used. But again, it's expensive. I would just suggest keeping it small scale and cost possible. So Jim's going to talk about digit droplet PCR.
Yeah, this is a technique that was developed a few miles from where I'm sitting right now by Biorad in Hercules, California. And this has emerged as the way to determine the number of viruses that you have. And as Maggie said, there are 2 real ways to describe the number of viruses. How much protein is there per ml, that is the capsids, or how much DNA is there per ml, how many viral genomes per ml. And the latter has emerged as the way to describe viruses. And so what you're probably used to and having your students do is to put the ingredients together for polymerase chain reaction and then run the thermocycler and have either a band if the target sequence is there, or no band if the target sequence is not there. What happens in digital droplet PCR is you take your reaction volume, and the machine uses ultrasound to break it up into about 20,000 droplets. Now, each one of those droplets now goes through PCR cycles. And if the droplet has a viral genome in it, that droplet will give you a positive. If the droplet does not end up with a viral genome in it, the droplet gives you a negative. And then what the machine does is it just counts the number of positive droplets versus the number of negative droplets. And if you can imagine, if you have a higher concentration, then you have more positive droplets than negative droplets. If you have a low concentration, you have relatively few positive droplets. And so this gives you a number, and you then have it validated for what result you get with a particular concentration of viral genomes. And again, it's pretty common to have 10 to the 13, 10 to the 14, 10 to the 15 viral genomes per milliliter in the final product. Next slide.
So analyzing the purity of AAV after chromatography, for example, in downstream processing, and traditionally it was done using SCS page and either chromatin staining, as you see here, or silver staining. And basically what we're looking for is the presence of the 3 capsid proteins, VP1, VP2, and VP3 from viral protein. And the molecular weights of these are 87 kilodaltons, 73 and 61 respectively. So these proteins exist in the capsid of the AAV in a ratio of 1 to 1 to 10. So this is a very typical pattern that you see in an STS page gel. And you see lots of sort of images like this around. So this is, these fractions here represent purified AAV2, serotype 2. This is not purified using affinity chromatography. Usually it's much cleaner than this, as you saw in a previous slide. This was done using an alternative to that, which maybe would be cheaper, which is a precipitation method. And I'm happy to provide that reference. They used a precipitation method to precipitate selectively the virus and got this level of purity, which isn't too bad. So typically, checking purity as far as protein contamination is done either by SDS Page or by capillary gel electrophoresis. Again, this is common in traditional biomanufacturing for purity analysis and presence of host cell proteins. It's again a denaturing electrophoresis. Proteins are denatured. They're separated on a very thin capillary gel and separated based on size, and the presence is picked up by a laser within the instrument. A big advantage of this is you can use even less material to analyze. It can be automated, and so multiple samples can be run at once. It has a higher sensitivity and also has the potential to fluorescently label the proteins and even have more sensitivity in detecting them. So this is what one of those units looks like. This is the sample preparation. It's injected into the instrument, and the readout looks more like this. So here you can see VP3 with a higher peak, VP2, and then VP1. Similar to traditional slab gels, the proteins are separated based on size, as you can see here. So that would be the readout of this type of analysis. So moving on to safety testing and also to environmental monitoring. And from what we learned from the industry subject matter experts was a lot of this is very similar to what exists right now with traditional biomanufacturing, which is great because we already teach this, except for some differences with the length of some of the traditional microbiological tests. So we'll talk about what's specific then for the needs of cell and gene therapy there. So when we think about microbiological control, we're really thinking about presence of mycoplasma contamination, endotoxin contamination, sterility tests, bioburden tests, and also environmental monitoring, because of course, all of this has been done in a clean room environment. So this is the compendial sterility test that's approved by the FDA. Jim, do you want to talk about this test?
Yes, so when you tell your students about the United States Pharmacopoeia, that has standard tests to do a variety of things, to show you the standard tests that will show you that salt is salt. How do you test for water sterility? You look at the US Pharmacopoeia. In US Pharmacopoeia 71, this is the gold standard of how you prove that any drug substance or any drug product is sterile and is worthy of being parenteral, of being injected directly into the bloodstream of children. And so the standard way to prove that your final product is sterile is to take a sample of it and to add it to two media, fluid thioglycolate media and really tryptone soy broth, a very, very rich media. Now, many of you are probably teaching microbiology and you probably use thioglycolate media to grow analytic bacteria. And so this is something you can bring into your microbiology class, that these two media are used and the two media are incubated for 14 days. They're incubated at two different temperatures, at a lower temperature that might favor the growth of yeast or other fungi, and a higher temperature that might favor the growth of bacteria. And in the end, it's a positive or a negative, just like in our microbiology class. Do you have turbidity, showing that your original sample was contaminated, or do you not? I've included here the 6 different standards that serve as a positive control. in a growth promotion test. You have to grow these, which demonstrate for every batch of media, which demonstrates that your media will show a positive if these bacteria or fungi are present. And you have a aerobic bacteria, you have anaerobes, you have gram positive, gram negative, you have a yeast, and you have a filamentous fungi. So all of those are grown as a positive control. And you incubate, as I said, you incubate these for 14 days. That's to catch slow growing bacteria like P. acnes that may be there and growing really slow. Now, the problem is when you're talking about cell therapy, you don't have 14 days. The patient's cells are coming in and you are on the clock. The patient has a month to live. It's urgent that you get this back. It's urgent that you demonstrate to the FDA that there aren't any bacteria and you're not going to inject bacteria into the patient along with their cells that have been sent to gene therapy boot camp, but you don't have the 14 days of the standard compendial test. And the FDA is allowing alternative methods, including the one that Maggie's about to talk about.
So this is one example of a rapid testing system for sterility. As Jim just said, especially with CAR T therapy, where the whole process takes 3 weeks, you can't wait 2 weeks before it gets injected back into the patient. And also these types of tests allow you to test a smaller sample. With CAR T therapy, for example, any sample you take from the production is cells that don't get to the patient. So you want smaller sample testing and you want it to be fast. So this is one system that's in use now, and it has been in use for a few years now. It's the automated BAC-T alert system from bioMérieux. These are the media bottles that get inoculated with a small sample, and they're placed in this instrument here. And what happens is if there's any growth, CO2 will be produced from bacterial or microbial growth. And right at the bottom of each of these is a substance that is pH sensitive. It will change color if the pH changes due to a CO2 production. And within the instruments, there's a laser then that continuously scans each of these bottles and looks for a change in this color. So it reads the samples. I think it goes around, does it every 10 minutes or so, such that if there's a positive result for any of them, that will be indicated in the instrument. It's very sensitive. Sensitivity is down to 1 to 2 colony forming units, and it's much faster. So this is really required, especially for CAR T therapy and other cell therapies, with a positive result being found in 1 to 2.5 days. So these systems have been validated with different types of bacteria and all the controls that Jim mentioned as well. So this is what is really needed to be used in productions for CAR T therapy, and is also used for gene therapy. In addition to sterility, we're also required to test for mycoplasma and endotoxins. So mycoplasma testing, just like in traditional biomanufacturing, is done using quantitative PCR, typically targeting the 16S ribosomal RNA gene of mycoplasma. And with this approach, more than 70 mycoplasma species can be detected with the one set of primers, with the one test. Again, for traditional more cell growth methods for detecting mycoplasma might take 21 to 28 days. And that's just not a possibility for these quick turnaround products. Endotoxin testing, very similar to traditional biomanufacturing, where it's the limulus amoebocyte lysate, LAL lysate. There's several different forms of that. There's a colorimetric test, a turbidity test, and also the traditional gel clot test that, if you don't have fancy equipment, this is a great test to introduce into your class. And then moving on to environmental monitoring, of course. So clean room monitoring, making sure clean rooms meet specifications in a scheduled manner with testing, but also testing and sampling during production. These areas, these clean room areas, are sampled for microbial content in the air through active and passive sampling. Active sampling involves something like this. This is a handheld air sampler where a cubic meter of air is pulled in through a metal sieve and onto a bacterial or microbial growth plate. And then it's incubated for 3 days to look for microbial growth. And then depending on the level of clean room, specifications are set for how many colony forming units can be present in that cubic meter of air. Passive air sampling too, using settle plates, where those are just large microbial growth plates that are left open for about 4 hours, and anything that is in the air will settle onto the plates. And again, they can be incubated and evaluated for growth. In addition to that, the number of particles in the air has to be sampled on a regular basis. And again, using specifications set for each of the different types or levels of clean room. The sampling will be done during production in biological safety cabinets in class 10,000, class 100,000. And those numbers represent the number of particles that are sort of allowed that meet the specification. It has to be under that amount to meet specifications for that particular clean room. So this is very similar to what's done in clean room monitoring for traditional biomanufacturing. In addition to that, there's surface monitoring with contact plates, swabbing of areas that are not flat. So there's an environmental monitoring protocol that has to be in place. There's a schedule that has to be in place. When microbes are found and that's out of specification for those areas, they have to be identified and then investigated. Where did that contamination, where did that microbe come from? What's the source? And that's often outsourced, as we learn from our subject matter experts. The identification of these microbes are outsourced, is outsourced. So again, with environmental monitoring, you are establishing a regular schedule of monitoring during certain times during the production, taking samples at different locations. And in addition to that, there's also a validated cleaning protocol that's in place for cleaning the clean rooms with appropriate disinfectants and appropriate scheduling of the cleaning as well. Again, this is very similar to traditional biomanufacturing. When the data is collected, of course, it is analyzed, and also the trends are analyzed for each of the clean rooms. So from what we know and from what we have from our subject matter experts, the environmental monitoring component is very similar to traditional biomanufacturing, but the rapid sterility tests are a must for mycoplasma, for sterility, for these advanced products.